Hi class, Dr. Jim here. In this lecture, we're going to be looking at the senses. So this is the second part to chapter 38. So the first part we looked at was the central and peripheral nervous system and really focused in on the parts of the brain and how it functions and what are the different parts do inside the brain. In this lecture, we're going to look at the specific senses. And so this is how we respond or what we get as stimuli so that we can respond later on. And so in chapter 39, we're going to look at how do we produce motor signals to respond to our senses that we see. So it's all kind of building up to the last part of responding in behavior to what we're stimulated by from the outside world. And so we're going to really take a look at all the five senses. We're going to talk about the different types of receptors we have. We're going to focus in a little bit on the taste buds, a little bit about the ear and equilibrium and how all that works, and then finish up looking at the idea of sight and what really happens when we see things through our eyes. And it's really our brain that sees. Our eyes are just receptors that see or that get stimulated by the light, and then that sends images to our brain, which puts it all together and actually sees the pictures. So it's our brain that sees, not so much our eyes in that, and we're going to talk about that as we go on. Okay, so that's kind of what we're going to look at today, is take a look at all five senses and see how they all work together so we can perceive our world. And so again, we're going to look at how does the sensory system conduct responses. So we'll talk about all the different ways of how the sense nerves sense and then send that message back to the central nervous system. We're then going to focus in on what are the different types of receptors that animals have to sense the world. So we'll talk about the five different types: the mechanical receptors, the electromagnetic receptors, the taste, the olfactory, and then we'll also look at the pain receptors as well. And so we're going to kind of focus in on all those different types of receptors that are out there and how they respond, or how we sense that and how that actually works. And then again, how we respond will be in the next chapter. Then we'll look at how does the hearing and equilibrium of mechanical receptors work. So we'll look at how in conjunction that the ears play a role not only in sensing sound, but also keeping us so that we avoid getting dizzy. So a lot of people don't realize this, but the ear is also responsible for keeping us straight and having our equilibrium right. And so when we have problems with dizziness, a lot of times you'll see people, especially on boats and that stuff, put these patches behind their ears, because what it does is release a chemical that will help stimulate or relax the equilibrium a little bit, and so they don't feel so bad and seasick on a boat. The last thing we're going to look at is then how the visual receptors work in animals. We'll talk a little bit about how the eye actually gets the receptors. We'll talk about the difference between cones and rods and what they do. I always remember cones see color, rods see light, and darkness. And so the rods are more for the light and dark perception, where the cones are necessary for the color. And so we'll talk a little bit about that. We'll look at the differences between animals that have many cones and many variety of cones so they can see multicolors versus those that can't see as many. And then we'll talk a little bit about color blindness at the very end, in case anyone out there is colorblind, or if you've heard about it and what does that all mean. So we'll look at that as well. Okay, so we're going to take a look at the five senses today. So the first thing we need to talk about is when a sensory receptor gets a stimulus. And so what it does is it changes the membrane potential and sends that message out to the central nervous system. And then the information in the central nervous system results in a sensation. And so the sense comes from one of these receptors out there. So the eyes, the nose, the tongue. Equilibrium or sound could be touch. So again, touch could be one, or sensation here. And all those messages travel up either up the spinal cord or the cranial nerves to the brain. And so the brain then perceives what it's sensing based on where these impulses are coming from. And so we'll look at that in just a second. Now, a sensory pathway begins with a sensory reception, and so it could either be by one of two things. So you can have a sensory receptor built into the neuron itself, and so the neuron is kind of the end point in this case, and it has the sensory receptor on it. Or there could be a sensory receptor cell that's connected to a neuron, and it sends a neurotransmitter across, and then that sends the signal. So that's the type of thing that we think of. So you either have a neuron that has sensory reception, or you have a sensory receptor that then sends that message to our neuron to cause a response. And so this is with both inside and outside the body that's going on in these cases. Now, sensory transduction is the conversion of a stimulus energy to membrane potential of sensory receptors. And so here's a great little example showing you. If you get a stimulus with a receptor cell, it sends out a chemical messenger, and that stimulates the open channels for sodium to come in. When the sodium comes in, that sends an action potential down the neuron. Now, in a cell that ends in a modified receptor, what happens is you get the stimulus. Now, it has, again, these signals that trigger the gated channels to open. The sodium comes in, and now you get an action potential down the neuron. And so that's what it's called. Now, when the membrane potential or the change in the membrane potential is called the receptor potential, and the receptor potentials are graded. Their magnitude varies with the strength of the stimulus. And so in the next slide, we're going to see how different senses or different magnitudes of sense will trigger either a graded response or additional responses in that. And so that's kind of how this works. But in order for these things to sense, they need to get some kind of stimulus in order to open up those channels or send a signal to open up those channels in these neurons. So the sensory information is transmitted by nerve impulses or action potentials. The neurons act directly as the sensory receptors produce action potentials. So that's in this case where the stimulus hits the end of the neuron and then opens the sodium channels right away. Or you have a sensory receptor cell which releases a neurotransmitter which now opens the sodium receptors. Non-neural sensory receptors form chemical synapses with those, and the typical response to stimuli by increasing the rate at which the sensory neurons produce action potentials. And so more stimulus means more reception in that case. And so that's kind of the idea. Now, the response of the sensory receptors varies with the intensity of the stimuli. So if you get gentle pressure, you get low frequencies of action potentials per receptor. So gentle pressure will only send small amounts of action potentials every so often. The more pressure you apply, the more frequency of action potentials you get. And again, that gets to a level to the point of where these neurons can only respond to, once they go to the repolarization and that. And so that spot where the repolarization, wherever they can trigger. But you can see the more pressure you apply to these sensory receptors, the higher frequency of waves you get as opposed to low pressure, which only sends occasional action potentials. And so you don't feel it as much. And so that's the idea. The more pressure, more sensitivity, the more action potentials, the more you're going to feel it. And so that's kind of how that all works. Now, perception is the brain's way of putting together stimuli. And what's interesting is the brain puts it together by basically the route at which the stimuli comes from. So if it's coming up from the spinal cord, it's probably meaning it's a sense from one of the limbs or the skin or somewhere else. That's going to be a somatic sense. If it's coming from the nose, it looks like it and it reads it as smell. If it comes from the eye, it's visual. From the tongue, it's taste. And sound, it's going to the lower part of the brain, the temporal lobe of the brain. And so really that's how the brain perceives these senses. If you could kind of mix up senses and that stuff if the brain perceived it was coming from the hearing and it was actually from the body and that's how these things are. It's really about how it arrives. It's not that the brain has to think about it and think, where is this sense coming from? What it does is it just basically says, oh, it's coming from the ears, then I need to respond to it. Or, oh, it's coming from the spinal cord, that must be a sense from the body. And then, oh, I see it from the visual. Okay, then I'm putting a picture together and I sense it from there. And so the brain just knows based on the pathway that it comes from. And so that's what it is. It doesn't have to think about where is the sense coming, where is the sense and what is it before it can respond. And so that's the idea, is it's basically using the pathway as its mode of information in that case. Now, with these senses, you do have amplification. So when you apply a stimulus, you can get these tonic receptors that will apply more action potentials. Okay, and so the more stimulus you get, the more action potentials you get along the way. And so that's one of the things. You also have sensory adaptation, which is a decrease in responsiveness. And so you get an action potential, but after a while it stops signaling. And that's kind of like what we have in our skin. It's like when we put on our clothes for the first time, we feel it, but then after a while we don't, and we don't feel it anymore because it's not really sending any action potentials from our neurons. We're adapting our senses to that. It's the same idea if we were washing our hands in warm water and we got them nice and warm, and then all of a sudden we stick them in the cold. What happens is that we still sense it as warm water. It's kind of weird, even in the cold water. And if we did the same thing with cold water and we turned it to hot, it actually is pretty chilly at first because our adaptation that we have, because our senses are sensing it, and it just assumes that's what's going on. And when you flip it right away, it takes time for that change to happen. Same thing with the phasic receptors. Is kind of like when we have things on our bodies, the pressure receptors kind of sensory adapt so that we don't feel it as much, and so we're not constantly getting bombarded by action potentials from our clothes or wearing a hat or wearing glasses or something else on our bodies. So we tend to forget these things are on us because we don't feel those pressure or senses all the time. Okay, and so that's what the sensory adaptation means, is that basically it puts it down so that you don't respond constantly to that same stimuli each time. All right, so based on the types of sensory receptors we have 5 different categories. We have the mechanical receptors, we have the electromagnetic receptors, the thermoreceptors, the pain receptors, and the chemoreceptors. So the chemoreceptors are going to be the smell and taste. The mechanical receptors are going to be the touch, the free innerving. The electromagnetic receptors are really the vision, but you could also say the equilibrium as well, because that's kind of electromagnetic type of thing. And then the thermoreceptors are also going to be in the touch as well as the pain receptors, these free nerve endings, and sometimes the Meissner corpuscles. And so again, some of these things have a couple of different functions, but depending on the type of sense, these are the 5 types of senses that we have and how we respond to them. Okay, so that's what we're going to look at. Now, mechanical receptors sense physical deformation caused by stimuli such as pressure, touch, stretch, motion, and sound. And so some animals use mechanical receptors to get a feel for their environment. And for example, the whiskers on both cats and rodents are very sensitive to the world around them. And so when they can sense where objects are by basically feeling the whiskers getting brushed or how close. And so a lot of times you'll see when you see a mouse come in the room, they kind of twitch their nose. And the reason for that, they're trying to sense where everything is. And they use their whiskers to kind of get a picture of where everything is. And so not only do they use their eyes, but they also use their whiskers as kind of a depth perception and kind of helping them out. And so that's one of the things. Now in humans, we have a couple of different things. We have the Merkel's disc, we have the Meissner's corpuscle, and we have the Krause-Zen bulb. And all these things in the Ruffini ending and the Peckian corpuscle, these are all mechanical receptors that we have in our skin, and again allowing us to sense the different pressure that's going on in the world. And so that's one of the things that we look at. Okay, now electromagnetic receptors include electromagnetic energy such as light, electricity, and magnetism. So this is our eye, our visual sight. But other animals use electrical receptors to detect an electrical field generated by their prey. So they can sense that in the water. Other animals may use this as a way to sense prey in that, where they can do temperature in that with the or electrical magnetic field. And even some animals use the magnetic fields to migrate, and so they know which way is north and which way is south based on the magnetic pole and the way that they feel, and so using that in those cases. Now another one is the thermoreceptors, and again these detect heat and cold. And again, some snakes have thermoreceptors that detect the body heat against prey in the colder background. So if we look at that, that's kind of near their nose, and that here's the nostrils, and they have these little organs here that detect heat, and so they can kind of see a heat image as well, not only with their eyes, but they also get a sense of heat. And where especially warm-blooded mammals are, because they give off a lot of heat, they can sense those and attack and go after it right away and see it without actually seeing it. They can feel it with their heat receptors. Now, in humans, thermoreceptors in the skin and anterior hypothalamus send information to the body's thermostat, the posterior thalamus. And so you can see how you have neurons that respond to heat, some that respond to cold, and some that are in the middle. And so there's the intermediate in that. And like I said, sometimes we can even confuse these receptors. what happens is we can be washing our hands with cold and then all of a sudden switch it to hot water right away, and it takes time for these to switch over. And so sometimes then you feel it's still cold even though it's hot water, and so it takes time for it to warm up, and that's due to the senses, and again switching over from the hot receptors to the cold receptors in those cases. Now, we also have pain receptors, and these are called nociceptors. And again, these detect stimuli that reflect conditions that could damage animal tissues. And so essentially, this is there to tell us this is bad. We need to move or respond to it. And so, a triggering defense reaction, such as withdrawal from danger, pain reception serves as an important function. And there are chemicals such as prostaglandins that worsen pain by increasing receptor sensitivity to noxious stimuli. There are things we take like aspirin and ibuprofen that reduce pain by inhibiting the synthesis of prostaglandins so we don't feel pain as much. And so it's not that the pain isn't there, but it makes our body not think that the pain is there. So we prohibit certain chemical mediators from telling us that the pain is there, or bad pain, and so we feel a little bit better when we take those things. And aspirin actually has some other qualities to it too, and I think I mentioned with blood coagulation and that as well. And so sometimes aspirin works also on, especially like headaches and that stuff, where sometimes that's due to vasoconstriction, and what it will do is actually vasodilate in those situations. And so some of that is also due to the role of aspirin and ibuprofen for the vasodilation of blood vessels. And so we'll see that as well. Okay. Now, the chemoreceptors that we have transmit information about the total solute concentration of a solution. The chemoreceptors respond to individual kinds of molecules. We also have olfaction and gustation that depend on chemoreceptors, and smell is the detection of odorants carried in the air, and taste is the detection of tastants that are present in solution. So we do have chemoreceptors in the body that will respond to conditions in our blood. And so we've talked about this before, where again the number of how much O2 or CO2 or the pH, and all that can be trigger a chemoreceptor in our body, where around the heart can increase the blood output and cardiac pressure and blood pressure due to vasoconstriction, which will then trigger the pH to go up, and then to raise the pH as well as the level of O2 and lower the level of CO2. And so that can happen in that case, and that would be a chemoreceptor where the pH level has gone down and then you have to stimulate that up. So that's a chemoreceptor in the body. Now, the ones that we're primarily familiar with are the taste buds that are found on the tongue, and we'll talk more about that. That's found in the papilla that are on the tongue. We also have the olfactory bulbs in the nose that are little hairs that get stimulated by odorants that are in the air. And again, we can sense what's in the air based on that. Now, the chemoreceptors, like I said, humans can distinguish thousands of different odors, and that's through the olfactory bulb and the olfactory lobe in the brain. And those are transmitted through the nose, those little fibers through the hairs in the nose. And again, the hairs pick up the sound and are able to sense that. Now, humans and other mammals can recognize just 5 types of taste: sweet, sour, salty, bitter, and umami. And umami is savory. That's kind of the fancy word for savory in this case. And so that's the other taste. And so it's one of those things that's kind of a mix between salty and kind of just that savory home good taste, I guess. I don't know, some of the spices and that you can kind of think of. That's umami in that case. Now, the taste receptors are organized into taste buds, and they're found in projections called papillae. And again, any region of the tongue can detect any of the 5 types of taste, and you can see how they're all arranged: sweet, salty, bitter, sour, and umami in this case. And again, the food molecules come in, they tickle the right taste bud, and then that sends a signal to your brain telling you what you're actually tasting. So whatever taste bud gets stimulated, that's what's going to be picked up. And then you say, ooh, candy is sweet, or fries are salty, or a potato chip salty, or sour, a lemon is real sour or bitter in that. And so that's how we detect these different things. Okay, so through our taste buds that are found on the papillae on our tongue. All right, now hearing and equilibrium are mechanical receptors that detect moving fluid and settling particles. And so hearing perception and the body's equilibrium are related in most animals, and we're going to look at that comparing an insect to a human and that. But even if you look at most mammals, the mammals have the hearing the same way, and we have an eardrum, we have the cochlea, and we have the semicircular canals, which are responsible for the equilibrium. And you can see very similar to what we see in a cat that we see in a human. And again, when we look at other mammals, it's going to be very similar. The only difference is maybe the size of the ear in catching those wavelengths and that stuff. But essentially, that's the hearing part, is catching it and then bouncing it off the eardrum and then stimulating that. The fluid in that, typically through the semilunar canals, these are going to sense equilibrium. And again, we're going to look at that later on, where you have these receptors that can sense when you're turning and twisting in the semicircular canals. And so we'll look at that as well. Okay. Now, most invertebrates sense gravity and maintain equilibrium using mechanical receptors located in organs called statocysts. And so essentially, it kind of looks like our semicircular canals, and we have this stratolith on the inside that bounce against the cilia, and that tells the animal which way is up. So it kind of rolls around, and it can kind of tell which way is up and upside down and that stuff, and so kind of detect those different things. Now, in insects, sense sounds through body hairs that vibrate at different frequencies, and others have membranes that detect sound by means of vibration of sensitive organs, and then they can sense the sounds. But like I said before, most of the organisms can use the statocyst to determine what is up, what is down, what is sideways and right and left, and all that good stuff through that these sensory organs. Now, in most terrestrial vertebrates, sensory organs for hearing and equilibrium are closely associated, and so you see the same type of membrane and semicircular canals in most of vertebrate animals. And again, the hairs in the mammalian hair cell are what respond to sound, and then inside the semicircular canals is what you see for the, again, the equilibrium, and we're going to talk about both these things. In the cochlea, that's going to be where you detect the sound. We're going to look at it precisely, but essentially what happens is the vibrations bounce off the tympanic membrane, which stimulates these three bones, the malleus, the incus, and the stapes, and that bounces off the oval window, which then sends vibrations into the cochlea, which the hairs will detect based on the vibrations, and then that will transmit the sound to the or the vibrations to the brain, which is then picked up by the brain and used as stimulation. And again, what will happen is that the sound will then go through to the round window, and that's where they dissipate at the round window. So you have the oval window where, again, the bone, the stapes bone, bounces against and sends the signal down the cochlea, and then you have the round window where essentially where the cochlea ends, and that's where the vibrations end as well. And so we'll take a look at that as we go along here in a few minutes. So vibrating objects create pressure waves in the air, which are transduced by the ear into nerve impulses perceived by sound as the brain. So in this situation, if you had your earbuds in, you have this sound wave that goes through and it hits the tympanic membrane here, the eardrum, and that causes the bones to vibrate into the cochlea. You also have the bone conduction pathway that passes through the skull and passes down and also sends signals to the cochlea. And the cochlea is then going to send those nerve impulses to the brain. So that's where it goes from a sound wave to a chemical or a action potential wave down the nerves. And so that's where those things are. The three bones of the middle ear transmit the vibrations into the oval window and the membrane on the cochlea surface. And so we'll talk about that here in a few minutes. But essentially, that's how the sound gets in. It goes into the ear canal, bounces off the tympanic membrane, causes the bones to interact and bounce off the oval window, which goes into the cochlea, which is then perceived in a chemical, and then the hairs vibrate, which then send the action potential to the nerves and then finally to the brain. So that's typically what happens. Now, vibrations in the bones in the middle ear create pressure. The fluid in the cochlea will travel through the vestibular canal, and that causes the hairs to move, and that will cause a neuromuscular not a neuromuscular, but a neurotransmitter to be released and send a signal down. And you can see here where the bending of the hairs in one direction sends many action potentials, but in this situation, bending of the hairs in the opposite direction lowers the action potential in this case. And so it's all due to these hair cells, and the hair cells open and close or close or cause the channels to open and close due to the auditory nerve sensations that the brain interprets the sound. And so again, it's these hair cells that trigger the chemical response and sends the action potential down the nerve cell. Okay, and so that's how that works. Now, like I said, the round or the oval window is where the bone bounces the message off, and it travels down and it hits these hair cells, and that's the vibration it feels. It continues and goes down this pathway until it reaches the round window, and that's the end of the auditory pathway. Now, it's interesting. So, why they say people lose their hearing over time is due to the hair cells. Now, the more vibrations you get, the more these hair cells respond. And the ones that we lose first are the ones closest to the oval window in this case. And so, what happens is because they get hit so many times over and over again that they degrade over time and they go away. And so the louder the music you get and that, the more vibrations this gets, the more damage it does to these hairs. And so the hairs here are the first ones lost. And so as you age, the more and more low sounds here you lose. And then the only hair cells that respond are the ones farther upstream because these have not been damaged as much. And so that's the other reason why over time you lose the ability to hear very low tones. And so again, you will test your ears, and so a kid can hear all these different things, but an adult can only hear probably from about here on, because again, the damage to those nerve cells, and those nerve cells do not come back. And so that's one of the issues with the sound. And so again, the dissipate, and when they strike, the nerve strikes at the end of the round window, and so the sound goes through, causes these bones to vibrate. You go through the oval window, the sound travels through, passes through here, and then dissipates through the round window. Now, like I said, these are the hair cells. The hair cells take the nerve endings, tickle the nerve endings, and then send that action potential down the cochlear nerve and send it to the brain. Okay, so that's how we hear in these situations. Now, the ear can capture information about volume by the amplitude of the sound wave and the pitch, which is the frequency of the sound wave. And the cochlea can distinguish pitch because of the basilar membrane that's not uniform along its length. And so, again, you have this membrane that's different, and so it can detect the pitch and the number of wavelengths that are going on, or the frequency of wavelengths based on how many are coming through, and that's due to the membrane that's along there. And each region of the basilar membrane is tuned to a particular vibration frequency, and like I said, that's the hair cells that are responding to that. And like I said, you lose the low tones first, and you keep the high tones because they're farther downstream of where the oval window is in that case. All right. Now, several organs in the inner ear detect body movement and position. Let's see if I can get this right. The utricle and the saccule contain granules called otoliths, and they allow us to perceive position relative to gravity and linear movement. And this is found in the semicircular canals. And so again, this is responsible for our, again, equilibrium. And so again, you have these hair cells that are surrounded by fluid, or this perilymph, and this perilymph will move in one direction or the other, and it will tell which way the body is moving in one way or the other based on this movement of fluid. And then that will send a signal down the vestibular nerve to the brain to perceive equilibrium. And so that's the one thing. And so again, these semicircular canals contain fluid and can detect the angular movement in any direction. And so this is what's responsible for keeping us from getting dizzy or falling over or doing those things, because it perceives us which way is the right way. And so when we move our head one way or the other way, we can perceive that based on the fluid in these semicircular canals. Now, the last thing we're going to look at is vision. And again, the diverse vision receptors in animals depends on the light absorbing pigments. And I'm just showing you here an example of the different cones that different animals have. And you can see some animals have 3, which we call trichrome, some have 2, some have 4, and others have variants based on again, some of the different receptors that they have. They call these beta receptors, and so they switch over, and so that's what the dotted lines mean. But you can see a goldfish actually can perceive more colors than even we can. Mouse only sees 2 colors, kind of like your dog and cat. A chicken can see 4. A salamander can see 3, but it also has these beta receptors that kind of also receive, and this is for the heat kind of thing, the infrared. And so that infrared is heat, and it can detect heat using its vision as well. And so that's where you see that. Same thing with the goldfish. Look at how many colors the goldfish can see, and lots of different things in that. Okay, so light detectors in animals range from simple clusters of cells that detect the direction and intensity of light to complex organs that form images. And all light detectors contain photoreceptors that contain light absorbing pigment molecules. And so, again, this is what humans have. We have blue cones, green cones, and red cones, and it's the combination of cones that get activated that allow us to perceive color. Now, where we see this is in the back of the eye. So again, we have this rod cell and cones. Cones see the color, rods see light versus dark. And so that's what you're seeing in these situations. And it's funny because light goes through this whole layer of tissue. The nerves are actually in the front of the vision, and then the cones are in the back of the eyeball. So if you look at this, the cones are actually positioned in the back, whereas the nerves are all in front of it. So the light actually has to go through the nerves and actually then receives the cones and rods, and then those get stimulated and then send the signal through the nerves, the action potentials through the nerves, which then go through the optic nerve to the brain, which then puts the picture together. Now, in most invertebrates have a light detecting organ, and again, it's the simplest of light detecting organs that are found in planarians, and they have ocelli, which are eye spots near the head. And so this just detects light versus dark and allows it to move away from the light and go to the darkness. So you can see here, they have this screening pigment, which can detect light versus dark. The visual pigment will pick that light up, it stimulates the nerves, and then it will pass it down and say, oh, that's light, I want to get away, and allow it to move away from the light. So it allows planarians to get away from the light and seek shaded locations to protect itself from predators. And so that's what it's doing there. Now, insects have compound eyes, and again, these compound eyes allow them to see movement very well. So that's why a fly can see you before you can come down fast enough to hit it. And so you got to be really, really fast. Otherwise, it can see you're moving already before you even make that movement down to slap it and try and kill it, because these eyes all see a different image, and each one of these, think of this as each one as an eye, and so they have eyes in front and back, and so they essentially see almost a 360-degree picture all around their head, and so they can detect and move it very quickly and so you have to be very, very fast to actually try and kill an insect because of the way that they can detect motion with these compound eyes. Now, most other animals like jellyfish, polychaetes, spiders, and mollusks have single lens eyes, and so they work on a camera-like principle where the iris changes diameter and the pupil to control how much light enters into the eye. And these show you some different examples of, again, the types of eyes that are out there. And all vertebrates have a single lens eye, and so the variants you see are in invertebrates. Most vertebrates, or all vertebrates, have single lens eyes if they have eyes. And again, that is all based on the lens, and these things allow for light to pass through and focus in on those different things depending on what type of eye that these guys have. And so you can see the wide variety of different eyes and the how these things develop over time. Okay, and so again, vision begins with the photons of light that enter the eye. They strike the rods and cones. However, it's the brain that sees. And so again, these detect the light and the color of light, but they don't put together the picture. It's not your eyes that actually see. They're just picking up the senses that you see, and then the brain is then putting it together and putting the picture together of what you see based on the cones that get rods and cones that get stimulated. So again, these are in the back of the eye. The light comes through, hits the back. The rods and cones get stimulated, and then they send action potentials. So they send out chemical messages to the bipolar cell, which then sends out chemical messages to the ganglion cell, which then sends it down the optic nerve. And you can see how this all moves through and then down the optic nerve itself. And so that's where those things go. Now, transduction of visual information in the nervous system begins when light induces the convection of the cisretinal to the transretinal. And so essentially what it does is it changes this retinal protein, or I wouldn't say it's even protein, but these monomers and that into the cis isomer to the trans isomer. So it changes this thing from this position to this position inside the rhodopsin in this case. And so again, in the cell membrane where you can see these discs, and that sends the signal out through these different things. And so again, you can see the difference between a rod. Rod is really long and a coat is really short and has these different discs. And these are the discs that respond due to the change in the retinal cis versus trans isomer. And so it goes back and forth, and that's what causes the sensation of light to be picked up and then send that message on to the brain. Now, when the cyclic GMP breaks down, the sodium channel is closed. This hyperpolarizes the cell, and then the pathway is shut off again as the enzyme convert the retinal back to the cis form. So you can see that light strikes the retina, you get hyperpolarization, and then you get the sodium coming in, which then will drive the system to send an action potential eventually as it goes along. Now, the process of visual information begins in the retina. In the dark, rods and cones release neurotransmitter glutamate into the synapses with neurons called bipolar cells. And these are the cells kind of in between the nerve cells and the rods and cones, these bipolar cells here. So essentially what happens in the dark, you get this neurotransmitter of glutamate in here, which stimulates the bipolar cell. And then they're hyperpolarized or depolarized in response to glutamate. And again, the rods typically see in the dark, whereas the cones do not. And so in the glutamate, that will be hyperpolarized for those bipolar cells. In the cones, they'll be depolarized so that you can see in the dark. And again, it takes time for the eyes to adjust in those cases. Now, in the light, the rods and cones hyperpolarize, shutting the release of glutamate. The bipolar cells either then are depolarized or hyperpolarized, depending on the sensation, and again, sending that out to the optic nerve. Now, the signals from the rods and cones can follow several pathways in the retina. Again, the single ganglion cell receives information from the array of cones, and the cones from each response to light from the perpendicular or particular location. The rods and cones that feed the information to one ganglia cell define the receptive field, the part of the visual field which the ganglia cell can respond, and the smaller receptive field typically results in the sharper image. And so that all this is saying is that you have a number of rods and cones that respond to the same bipolar cells. So you can see how they're connected to the same bipolar cells, and then those are connected to the ganglia. The sharper the image means that the fewer rods and cones that actually get stimulated, you get a better image. When you stimulate more rods and cones, it gives a brighter image, and those don't send out as nearly a sharp of signal as do if you only had a very specific set of rods and cones that actually get stimulated at a time. And so that's what it means. The sharper the image is going to be the fewer rods and cones that you receive images and send that out. So again, you see just in this drawing here is lights coming in through here. It goes in, it tickles the rods and cones, and then they send a message to the bipolar cells, which is an action potential, and then they send it through to the ganglion cells, which then send the message up the action potential to the again, the optic nerve. And so that's how it's going. So light's coming in, tickling these guys, sending a signal, and then these cells get activated and then send that message up through the ganglia, and again, through the action potentials that occur. Okay, so that's kind of how the visual works. It goes back, hits this part, tickles these guys. These send out chemical messages to the bipolar cells. Bipolar cells then activate the ganglion cells, and that sends out the message. And depending on how many of these get activated at once, determines how crisp an image actually is in these situations. Okay, now the optic nerves meet at the optic chiasm near the cerebral cortex. So they do cross, and where those optic nerves cross one another is where the chiasm is. And so again, the sensations from the left visual field of both eyes are transmitted to the right side of the brain. So the left eye will go over here, and the left side of this eye will go over here. And you can see that then it stimulates in the right brain. Whereas on this side, you have the right visual field, and that will go over to the left side on both eyes. And so the left side goes to the right, and the right side goes to the left. And so those are all the different things. And again, the sensation of the right visual field are transmitted to the left side of the brain, and it's estimated that about 30% of the cerebral cortex, so that's the back of the brain, the visual cortex, takes part in formulating what we actually see. So when you're visualizing stuff, you're seeing your brain is using about 30% of that visual cortex to put those pictures together. And so That's a lot of work for that brain to do. And so in that back of the head right here, so the back spot of your head right in the back there is where your vision is coming together and putting the visual together. So a lot of times people, if they hit the back of their head, will actually lose their vision because that's the part that actually sees, and you're using that part of the brain. So if you get a contusion on the back of the head, a lot of times you'll lose your sense of sight for a while because of where you got hit in the back of the head. And so again, that can be an issue, and you may never gain that sight due to that. But sometimes once it recovers and the brain recovers, then you may be able to perceive that sight again, depending on how damaged that is. Now, color vision among vertebrates is most in fish, and that's probably due to the dark water in that, so it can see around it. Amphibians and reptiles and birds all have very good color vision, and again, that might explain why you see such bright and brilliant colors in the males, because again, they're trying to attract the females, and so if they can see really good colors, that's why you see all those wonderful colors produced by the birds themselves. Now, humans and primates are a minority of mammals that actually can see color well. Most mammals only can see two colors, and so mammals that are nocturnal have a usually usually have a high proportion of rods rather than cones, and so that's why they don't see color as well. And so most mammals tend to only see two colors, and so you can see the difference between a dog and a human. Humans, we can perceive red and orange and yellow and green and blue and violet, whereas dogs can pretty much see yellow, a little bit of brown, and blue. And so this is kind of cool just to see what you see. You see all these colors. And so when they make toys, they make it for humans, not for dogs, because mostly dogs can only see yellow and blue, and that's all they're seeing. So if they see blue, or if you see blue, they're gonna be able to see that. But everything else is gonna look yellow or dark to them, and so they really don't see it. So the colors that you're buying for your dog are actually for you to see, not what your dog sees. And that's kind of funny because they make all these different colors and the dogs really can't see them. But it is a misnomer that dogs just only see black and white. That's not true. They actually see blue and they do see yellow. So they see two colors, but blue and yellow and kind of brown and that. So they don't have great color vision, but they do have some color vision. That's the same for cats as well. So if you have a cat at home, they see blues and yellows, but they really don't see reds and oranges and purples at all in that case. Okay, now in humans, the perception of color is based on three types of cones. And again, it's that arrangement of red, green, and blue. It's the same way your TV works. It's all these different colors. So now the LEDs have the three colors. And what they do is it's the number of colors that are being put together that allow you to see the different colors. And so again, that's what you see. You see the blue, the greens, and the reds. The rod is the black. And so again, you see light versus dark with the rods, and so that helps you see at night. And again, that's why nocturnal animals will have more rods, because they're going to see again, they want to see more at night when light is scarce, so they can pick up as much light as possible. Again, these photosins are formed in the retinal, or when the retinal binds to the three distinct opsin proteins. And so again, the colors that you see, and it's the combination of these colors allows us to see the other colors of the rainbow as well. And so again, the combinations of blue, green, and red allow us to see these other colors as well. Now, in some humans, again, we can have mutations which don't allow us to see certain colors. And so this is known as either abnormal color vision or color blindness. And so this is the normal, where we can see all the different colors of the rainbow. But if you're missing the rod cones, now you lack the ability to see really the reds. And so it looks more brown to people. And the greens don't work as well either. And that's the most common type of color blindness, is the red-green color blindness. Now, when the green cones are defective, that's known as deuteranope. And again, in this case, now the reds and the greens are missing, and the yellows are kind of blending, and you don't even see the brown colors in that, but blues are still. And then the trinope in this case is where the blue cones are defective, and now you can see reds, but you can't really see blues. They kind of just look grays in that. And so in this case, all you see is one color. In this case is the reds, and then dark for the rest of it. And so just kind of showing you some of the different types of color blindness out there. And there are a few in rare occasions have no color sense whatsoever, and they only see in black and white. And so there are situations where they're actually missing all three of the cones and they don't see any of the colors whatsoever. And so this is an X-linked gene, meaning that you're more likely to be colorblind when you're a male. That means if you get a bad copy of the X gene, in males, all you have is one copy of the X gene, and that the rest and those genes that are missing on the Y are found only on the X. And so if you get a bad copy on the X, that means you're stuck with the color blindness in that situation. Now in females, you have two X's, so if you have one bad copy, you always have a fallback in there. And the only way that females can be colorblind is if they have mutations on both the X genes. And so that's where you get color blindness in females. So it's much more rare to see a female colorblind compared to a male, which only has one X. And if he's colorblind on that gene, then he's going to be colorblind because the Y is missing that set of genes that respond to the rod color in that. And like I said, most common type is the lack of the red cone. So you get this red, lack of red in these situations. So you only see the blues, and that's really about it, and maybe a little bit of the yellows in these situations. Okay, so that kind of describes the color vision and what you see. So like I said, most people see this, but in color blindness, depending on what you're missing, what cones you're missing in these situations, really plays havoc on what colors you're going to see out or perceive out in the world there. Okay? So the visual field is the brain process the visual information and controls what is information is being captured. And focusing occurs by changing the shape of the lens. And so again, we have these muscles here that allow for the changing of the lens, and that allows us to fix in the focal image. Now, where light directly comes from is called the fovea, which is the center of the visual field. And that's the spot right here. You can see this is right in the center of the lens and where light is hit. This is going to have a large area of high density of cones, but no rods. So it's not going to detect light at all, but what it's going to detect, or not necessarily light, but it's going to detect the colors very well. So this fovea right here. And that's the center of the visual field in this case. And you can kind of see how this all works. So the center visual field is going to be where the fovea, and you can see right in the middle of that visual field. So that's going to really help you perceive color. So it's right in the center of your visual field. Everything else hits the different parts of the eye, and you can see how this all interacts with one another in these situations. So the green versus the red in those situations there. And you can see the green, which is on the left side, all goes to the right side of the brain, where the red on the right side now goes to the left side, and that's where you perceive it there. And the fovea, which is in the middle, is on both, where again you can see the difference, and that's where you split the right and left side. So that's the center of those of the visual field. Okay, and so that's how you perceive it. And again, the image itself, or the wavelength, is where this comes in. And where you have issues like visual problems, like as you get older and that stuff, or if you have visual problems, is the inability of the lens to actually focus the light properly and hit the right spot of the eye. And so it either focuses it too far, so that the focus is would be too far back of the image, or it's too close in the eyeball, and so you either have farsightedness or nearsightedness based on where it focuses in. And I didn't talk anything about that, but that's kind of the other issue as you age, is that these muscles don't respond as well. They get old and they get tired, and so the lens doesn't focus as well. And so typically people develop near or farsightedness when they get older if they have good vision, because again, they have a trouble reading and focusing in real close, because that image is now not being able to focus at the right spot properly on their eye. So they get glasses like this that allow them to see and be able to read really well, because it helps focus the image to the right spot on the eye, and so that the eye can see it clearly and get the right image that they're supposed to see. Okay, so that's looking at the eye and how the eye actually sees images in these cases. So we made it to the end of senses, and so I went quickly through this, and we're going to do a little bit more in the lab. I'm going to break out the sensory stuff. We'll have some fun playing with some of the different senses and kind of testing the senses. We have a thing for taste and smell, so we can test that. We also have some senses for touch, and then looking at reflexes and that, and then we can even check our vision and see how well our vision is in the lab and look at that. And so again, when we look at these things, sensory receptors transduce stimulus energy and transmit signals to the central nervous system. So again, you have the two types. You have either the neurons that have the sensory receptor built in, and that's the end of the neuron that sends the action potential, or you have sensory cells that then send chemical messages to the neurons through the synapse and then send the action potential to the brain. The brain basically realizes what the sense is by what pathway that it comes through. And so if it's coming up from the spinal cord, it's feeling the senses of the body. It's coming from the optic nerve, it's sensing that it's vision. Through the cochlea nerve is going to be the nerve of hearing. You also have the vestibular nerve, which is for equilibrium, and then the olfactory versus the taste buds in the tongue. Those are going to be able to do taste and smell. And so again, those are how we perceive those things. We've talked about the different receptors, the mechanical receptors, the thermoreceptors, electromagnetic, and the nociceptors. This is again sensing of sense, so pressure, and that for the mechanical receptors, thermoreceptors sense temperature, chemoreceptors smell, taste, and sound. Electromagnetic is going to be for vision and hearing, and that those waves, the different types of waves, the vision, hearing, and equilibrium. And then the nociceptors are for pain. And so that's what we see there. And typically those are just open end nerves that feel pressure and pain. And that's a evolutionary response to damage to tissue to say, you need to do something. You got hurt, go do something, either go away and recover or fix the wound or do something to it and do those things. Now, we also spent a little bit of time talking about hearing and equilibrium, and in most animals, these are pretty closely related. In all vertebrates, this is very closely related and typically the same organ. And so, especially in mammals and birds and in reptiles, the hearing and equilibrium are associated with the same types of organs where you have membranes that send the waves through the cochlea, and then the cochlea sends the information to the brain. And then equilibrium is the moving fluid. We have it in the semicircular canals. Other organisms have something very similar that allow for, again, sensing of what is upright in equilibrium based on the little otoliths or statoliths inside the little particles that move and respond to the cilia and trigger the cilia to make sure that they're standing upright. It's kind of like I'm trying to think of how you would think of it. It's kind of, if you think of like a pennies in a can, or beans that are being tossed around in something that's spinning, and the beans always stay at the bottom. That's kind of how these receptors are, that they're going to fall and trigger everything to make sure that you stay in a nice equilibrium. Now, when you spin it too fast, those things can't catch up, and that's how we get dizzy when we spin too fast in those cases. And so in order to maintain equilibrium, it's got to spin just fast enough so that these things can maintain the balance. And that's kind of how that all works in those situations. And then the last part we talked about was the diverse visual receptors of animals, and depending on the light absorbing pigments. I showed you how that kind of works. I didn't explain it as well as I probably could have, but essentially what happens is the light passes through, it hits the rods and cones. They get stimulated to release chemicals, which hits those bipolar cells. The bipolar cells then send action potentials to the ganglion nerves, which then send the signal on. And again, the closer or the smaller the signal of the rods and cones, the sharper the image is going to be. And so we looked at that as well. We talked about the chemical pigments and how well certain people can see, or not people, but animals can see. Again, most, I wouldn't say smaller, but most vertebrates can see color pretty well unless they're nocturnal. Then they're going to have more rods. And so a lot of mammals that are especially nocturnal don't see color as well as some of the birds and reptiles and that. And that's due to, again, being nocturnal, staying out at night. And so they have more rods than cones there. And you saw that like dogs and cats and a lot of the nocturnals only see two colors. And so they only perceive maybe blue and a little bit of green or red. And so they can detect that, but they don't see it very well. And so they don't see colors very well. Now, in humans, we see three colors: the rods, the reds, the greens, and blues. And again, it's the mixing of those that allow us to see the various colors. So we can see pretty well all spectrums, but there's some animals that even see more colors and can see heat as well using the thermoreceptors and the red light and shifting over into the heat infrared areas, which is the heat part of light. Okay, so with that, we've come to the end of the lecture. If you have any questions, please let me know. And that, like I said, we'll play around with the senses in the lab. I think it's always a fun lab to kind of screw around with. And if we're not too busy, we'll get all that stuff out and we'll play and mess around with our senses and do some fun things with that and play with that. And so until then, if you do have any questions, please send me an email or ask me when we get together. Otherwise, thanks for watching, and I'll see you next time.